Assalamualaikum everyone. In the today's video, we are going to discuss an important practical of biochemistry, and this practical is about detection of carbohydrate by Seliwanoff's test. In the previous lecture, we have discussed the practical about the detection of carbohydrate by barfoid test, but in this experiment, we are going to discuss Seliwanoff's test. So first we discuss the principle of a DAT test. That this test is used to differentiation between the aldoses and ketosis. You know very well aldoses are the sugar which has aldehyde group while the ketosis has ketones group in them. So the aldohexoses produce a light pink color after some times, while the ketosis produce dark red, deep cherry red color. So the difference between the aldosis and ketosis is of color, and the second difference is of the time. So the however after the prolonged boiling, the colors deepens and so restrict the prolonged boiling. So this test is based on the fact that carbohydrates are heated and ketones react more rapidly, and they dehydrate more rapidly than the aldoses. So the ketone, for example, ketone will undergo this test within the 5 minutes, but the doses can take more than 5 minutes. So this is the difference. So always keep the boiling for 30 seconds, not more than 30 seconds, because you know very well whenever we prolong the boiling, the color will be darkened. So in aldoses case, light pink color appear, but if you prolong the boiling, then this color will be dark into some dark red color. So therefore you should always have to boil for the 30 seconds. Ketones is react more rapidly with this Seliwanoff's reagent, because but why it is reacting more aggressive than the aldoses? Because the ketosis has 3 carbon atoms, while the aldehyde has only 2 hydrogen and 1 carbon atom. So therefore ketosis react more rapidly than the aldoses. So in concentrated HCl की presence में ketosis undergo dehydration. They will form furfural derivative, and they give a complex with the resinol, which is a alcohol, and give the deep cherry red color. So this salmonine test is very similar to the Mollis test, but Mollis test is for the all carbohydrates, but this salmonine test is creating the difference between the aldoses and ketosis. So it is very similar to the Mollis test, because in Mollis test instead of resinol we use alpha naphthol, and while the other all the things are exactly same. Now we move on to the next page. So you have to take a fructose and react with HCl. It will go under the furfural derivative, which is hydroxymethyl furfural. Then this again react with the resinol and give the cherry red color complex. Now first you have to prepare your reagent, and you will prepare your reagent by the resinol. You will take 0.5 gram of resinol and add distilled water and concentrated HCl into it. Now your reagent is prepared. Now procedure is that you will take 3 ml of that reagent and 1 ml of your carbohydrate solution, mix them and then boil for the 30 seconds. And after boiling you have to note the color which appears in the test tube. If the dark cherry red color or pink color appear, then you will indicated that it is ketosis or hexosis, aldoses or whatever. So at the last you will give the precautions you will take, that you should not increase the concentration of HCl, because whenever the concentration of HCl exceeded more than 12%, then it will can create complexes for us. So therefore you will have to take specific amount at the specific condition. So whenever you take more concentrated HCl, then it will convert your aldose sugar into the ketosis. So this will give the fast test. So therefore you have to take care about that. This is the equation which representing that test. So this is your fructose sugar, and it is going undergo dehydration in the presence of that reagent, and then it will react with resinol. This is the formula of resinol, and it give furfural derivative complex, and this complex color is of dark cherry red. so you can remember its name. First you have to draw simple sugar, and then you have to add resinol into it, and after resinol, three water molecules again dehydrated back, and this complex structure is obtained, and this complex structure will give its color, and then we can easily identify that what type of sugar it is. So in the second thing, you have to draw the diagram of test tubes. You will see that here this test tube keto sugar is represented because it has cherry red color, but in that you will given the light pink color ring appear, so it indicated that it is a aldose sugar. So that was all about the today's lecture. I hope कि आपको आज का लेक्चर समझ में आया होगा. अगर आपको फिर भी कोई कन्फ्यूजन है तो आप मुझसे कमेंट सेक्शन में पूछ सकते हैं, मैं आपको उसको ज़रूर आंसर दूंगी. सो टेक केयर ऑफ़ योर सेल्फ. मिलेंगे अगली वीडियो में, तब तक के लिए अल्लाह हाफिज़.